Good morning, everyone. Let me thank Professor Venkateshwarlu and Professor Kripanidhi sir for giving me an opportunity to talk to you in this conference. I am going to talk about some of the drug design strategies for the virus SARS-CoV-2, both based on ligand-based as well as target-based or structure-based. Um, so as you know, this is caused by the virus called SARS-CoV-2. So I will explain a little bit later on what is SARS-CoV and what is SARS-CoV-2. It affects the upper respiratory
Sebagian
tract, lower
kan kita
respiratory
sebagai
tract,
panitia.
and so on.
Iya, bisa
There are two
begitu
types
kan.
of SARS-CoV-2 strains, the S type and the L type. You must have heard some of them are more virulent and some of them are less virulent. So there are two types. So there are certain differences in their genetics. So if you look at a typical coronavirus, it's called corona because it's got like a crown-like structure. There are spikes, and this spike plays a very important role. It's called the spike protein, which goes and binds to the host system. So that is the first step in the process of disease. So the attachment of the spike protein to the host system, there is something called the angiotensin converting enzyme 2, the ACE2, which is found in the host, and the spike protein goes and binds to that. So, coronaviruses lead to cold and, of course, more severe diseases like MERS, Middle East Respiratory Syndrome, and then the SARS, the severe acute respiratory syndromes. Okay, so it can start from a very simple disease like cold, flu, going right up to the current pandemic. So there are 7 types of coronaviruses. The 4 ones, they are supposed to have been originated from bat. So if you look at the coronavirus, they have these spike protein. That's why it's called a crown. So these spike proteins play an important role in binding to the host with through a protein called angiotensin converting enzyme 2, ACE2. Okay, so they play a very important role in the start of the disease. So there are 7 types of coronaviruses. The 4 ones are much less harmful, leading to cold, flu, and so on. And the other three are the SARS, MERS, and the current SARS-CoV-2. So there are these two are called the alpha coronaviruses, and the rest of them are called beta coronaviruses. So the MERS, which is called the Middle East Respiratory Syndrome. Then we had the SARS, which is the severe acute respiratory syndrome, and the current one, which is the SARS-CoV-2. So this SARS-CoV-2 is more closer to the SARS-CoV. So these are much more virulent when compared to the other 4 coronaviruses. So if you look at historically how they originated, 1966, the first bat-based coronavirus 229E, which leads to cold, was discovered. And then, as you can see, the other coronaviruses, and then the outbreak of SARS in 2002, and then the SARS-CoV, the virus was discovered. And then in 2012, we had the MERS coronavirus, and then came the SARS-CoV-2, that is the current virus. So it's been there for a very long time, and the more virulent strains have started coming up in the past after 2000, I would say, actually. Okay, so typically virus is not like your bacteria or the fungus or yeast. They are very, very simple systems, and they have a nucleic acid, either RNA or a DNA, but not both. They have a protein coat, which encases the nucleic acid. Okay, then they also have a lipid or a fat layer outside, which covers the entire virus and keeps it more hydrophobic. So that's more like a protective. So it's very, very simple one. And so it cannot replicate or divide like your bacteria, making use of the carbon and nitrogen source available in the environment. So only when it enters the host system, it can start multiplying, making use of the host mechanism. Okay, so when it is outside, it's called virion, and its infective form. Okay. So typical structure of coronaviruses, the diameter is about 75 to 160 nanometers, whereas if you remember, bacterial systems are going right up to 1.5 micron. So these are almost, I would say, 100 times smaller than that actually. So the genome is a very simple, continuous, linear, single-stranded RNA. Molecular weight is about 5.5 to 6 um, 10 power 6 Dalton. Okay, so this is the and it's more spherical in shape. So if you look at the structure, we have the spike protein here, which goes, as I said, goes and binds to your host. Okay, then we have the envelope membrane protein. Okay, then we have another small envelope membrane protein, which then we have the your RNA inside actually, and then there is a lipid bilayer. So this is the constituents of the typical coronavirus. Okay, so we have the lipid layer outside, then we have the spike here, then we have the membrane protein, and then we have the envelope protein, which encompasses your RNA. So this current SARS-CoV-2 belongs to the beta coronaviruses. Okay, and interestingly, if you look at the sequence alignment of SARS-CoV-2 with the SARS-CoV, it's only 70% sequence similarity and 40% sequence similarity with the MERS. But this is taken as more closer to SARS-CoV rather than the remaining bat ones. Although genome-wise, they are much closer to the other four bat coronaviruses, but at protein level and as the mechanism level, it's more closer to SARS-CoV. That's why most of the homology modeling which we do to take protein templates, it's taken from SARS-CoV and not from the other four bat coronaviruses. So we have once more the spike protein, which is outside spike protein that helps in viral attachment, and then we have the E protein, that is the envelope protein. Okay, then we have the membrane protein, which encompasses your RNA, and inside we have the 3CL protease. This is more like a chymotrypsin-like protein. Then PL protease. This is more like a papain-like protein. And then we have the replicase, which is the important replicating protein, which takes part in replicating the RNA in the host system. So these are possible targets if one is looking at drug discovery. The S protein, I will talk about each one of them and how, what type of current drugs available, and how we can think about drug discovery as well actually. Okay, so these the ones here, the 3CL protease, PL protease, replicase, these are proteases type of membrane. Some of them are chymotrypsin type, some of them are papain like, some of them are RNA polymerase type actually. So like I said, if you look at the genome identity, the coronavirus is much closer to the bat genomes, 87%, and it's only 79% with SARS-CoV and only 50%, 49 with MERS. But it's taken more closer to this because at protein level and as well as mechanism level, it's more performing with SARS rather than here actually. So the data available, the protein targets available from SARS-CoV are taken for discovering drugs as of now for SARS-CoV-2 also. So if you look at the proteins, the PL protease, which I talked about, CL protease, replicase, helicase, so this is about 30 kilobase pairs in length actually. Then we have the spike protein, membrane protein, MNF protein, okay, here. So let's go into each one of them in more detail. Spike protein. Spike protein has three segments. So spike is like a cloud-shaped trimer. So it's got two subunits, the S1 and the S2. So S1 is the one, the head, and S2 is the stalk or the tail. So the spike protein, the S1 head goes and binds to the host ACE2 protein. Okay, the S1 goes and binds to the ACE2, whereas the S2 is fused to the RNA of the virus, which is going to enter into the host. So S1 binds to the host cell surface, that's the ACE2. Then S2 fuses the host and viral membranes. So the viral membranes enter the host cells. So that is how it looks like. So you have the S1 and S2 subunits. S1 is a trimer. I'll show you the PDB structure of that. Okay. And the S2 actually has the viral membrane which enters into the host. So this is the initial part of the virulence process. So this is the structure, the PDB structure. There are some PDB structures available for the current SARS-CoV-2 protein, but not many. As time goes, I think in the next 1 or 2 years, you may find many crystal structures in the databases. I have given here the PDB IDs here. So if one is interested in designing, well, you can download these PDB structures and design inhibitors to block the action. This C6VXX is the structure of SARS-CoV-2 spike glycoprotein. It's a closed form. That means it does not have the ACE2, whereas XLZG has both the spike protein bound to ACE2 here. Okay, so it's more of complex. So if you are interested, you can remove the spike protein and try to inhibit the ACE2 by designing new inhibitors. So you could do a lot of bioinformatics once you download these structures. Okay, so let's start each one of them. The S protein, like I said, the spike protein, it attaches to the ACE2. Okay, what is this ACE2? ACE2 is an angiotensin converting enzyme 2. So what it does is it converts angiotensin 2 to angiotensin 1 to 7. So it plays a very important role because accumulation of angiotensin 2 leads to hypertension. So this ACE2 converts this and reduces it to non-toxic products actually. So it plays a very important role in human actually. So ACE2. ACE2 is found in patients who have hypertension. ACE2, a lot of ACE2 is found. That's why you must have read in newspaper the comorbidity part. Those who have hypertension with coronavirus are more seriously prone to serious conditions. That's because they have a lot of ACE2. ACE2 is also found in lungs and many other parts of the body, much more than in several other parts of the body. So the spike protein goes and binds to the ACE2. This is again a PDB structure, 6MOJ, as you can see here. This is the ACE2. This is your spike protein. This is the receptor binding region. So you can prevent this binding if you can find out inhibitors which can dock and inhibit the process actually. Okay. Okay, once it goes and attaches, so we have the spike protein contains the S1 and the S2. So S1, S2 subunits. So there is another protease which is found in the host. It's called TMPRSS2. This protease cleaves S1 and S2. Okay, it breaks the S1 and S2. Now S2 is connected to the viral RNA which has to enter the host. Okay, through something called clathrin dependent endocytosis. So we can again design inhibitors to inhibit this particular protein TMPRSS2 so that it does not cleave the S1 and S2. So that opportunity is also there. Again, this is an endocytosis of the S2 RNA fused region into the host, so which is through clathrin dependent endocytosis. So we can design inhibitors to prevent this process also. Okay. Again, you can see the ACE2 receptor, and then we have the spike protein here. So the TMPRSS is a serine protease which cleaves S1, S2 and allows the S2 to enter the host, and S2 is fused to the viral RNA. So the viral RNA will enter the host. So the disease would have started actually. So again, these could be your possible targets. So once the viral RNA enters the host, there are other proteins like 3CL, PLP, then your replicase. So all these proteins replicates this. There is a proteolysis taking place of the viral RNA. Then there is a replication taking place. Okay. Now it's got many components which gets packaged and then there is an exocytosis, and the virus multiplied is again sent out actually. So there are many other proteins which take part in the replication, the 3CLP and the PLP and the replicase. So again, these could be possible targets for designing drugs. So once the viral proteins get replicated, they are packaged. You get more virion particles which exit the host and then exit as exocytosis. Okay? So the viral replication and exiting happen. So we have many possible targets as of now. But as I said, time goes on, when we come to know, researchers come to know more about the SARS-CoV-2, they may be able to find many more possible targets. Okay, so the proteolysis is done by the 3CL protease and PL protease. So these could be targets. Then we have the replicase, which replicates the viral RNA. Again, this could be another target for preventing the progression of the viral process actually. So many targets now is known. So we can look at the S protein receptor binding domain, because S protein, which is a part of the virus, the S1, which goes and binds to the ACE2. So these could be targets for designing drugs. Then we have the 3CL protease, PL protease, which are involved in the replication of the virus, the RNA. Then we have the TMPRSS2, which cleaves the S1, S2, so that S2 with the viral assembly can enter the host. Okay, then you have the cathepsin, through which the endocytosis takes place. Then we have the viral RNA polymerase that's in the pathogen side. So many targets have been, as of now, identified, and some of them have crystal structures available in protein data bank, some of them don't have. So we can do something called homology modeling from the SARS-CoV and then start looking at possible candidates. Okay. Okay, there is a web server if somebody is interested. There is a web server called https d3pharma.com. It gives you almost 77 targets for SARS-CoV-2 based on SARS-CoV. Many targets, as I said, do not have 3D crystal structure. So we may have to take homology modeling. So what you can do is, given an inhibitor, it binds to all these targets. Here I've written 42, but it keeps on increasing as time goes. So it binds to all the nCoV possible targets and then arranges them, ranks them based on which is the best target based on the binding energy. It also docks to human targets as well. So we can also find out if there are any side effects if the molecule which you have designed goes and binds to the human targets also. So this web server is useful if you are interested in looking at some possible inhibitors for designing towards this disease. It also does, given a molecule, query molecule, it can, using some fingerprint based approach, that means based on the structure, it can predict what could be its possible target. So this web server does a lot of things. I will show you at the end of my talk a couple of examples of what we have done with one or two molecules actually. So if one is interested in doing some bioinformatics, staying at home, you can use this web server and try to query with several hundreds of molecules, and it can give you what could be the possible best targets. Okay, so we'll go back again to the spike protein and ACE2 interaction. You can see this. We have the spike protein here. We have the S1 here. We have the S2 here. S1, it's a trimer. It's like a cloud, and then this is the tail S2. This also three stalks. You'd say this is the receptor binding domain. Then we have the ACE2 angiotensin converting enzyme 2, which converts angiotensin 2 to other harmless products. Okay, there are some ACE2 inhibitors reported here, okay, with the IC50 values. Okay, so there are some ACE2 inhibitors. That means they will go and bind to the receptor binding domain, so they prevent the spike protein attachment. But some important point you need to keep in mind: If ACE2 is inhibited too much, as I said, ACE2 plays a very important role in converting angiotensin 2 to much harmless product. Too much of angiotensin 2 could lead to cardiovascular hypertension. So one needs to keep those points in mind, especially those who have cardiovascular issues will also have the side effects related to this. But one can look at these, one can design structures, novel structures based on the structural similarity of these molecules. If you look at TMPRSS, like I said, is involved in cleaving the S1 and S2, two subunits, so that S2 with the RNA fused will enter the host. There are again some molecules that have been reported in literature, okay, with their IC50 values given here actually. So we can do something called a Pharmacopor based approach. So if you superimpose all these inhibitors, the three-dimensional structure of all these inhibitors one on top of another, and find out what are the common features. It's called pharmacopores. What are the common pharmacopores that they all possess? That structural features they all possess. A pharmacopore is a hydrogen bond donor like OH or NH, a hydrogen bond acceptor like nitrogen or oxygen, an aromatic group, a hydrophobic group like CH3 or C2H5, or cation or anion and so on. So if you develop a pharmacophore with known inhibitors for TMPRSS, we find interestingly a common feature: two acceptors and one aromatic. Two acceptors means either two nitrogens or two oxygens present, and there is an aromatic. Okay, so at certain distance. So that's the common pharmacophore for all these molecules. One aromatic, two acceptors. So what's the use of this pharmacophore? We can look at databases, structural databases, and identify or shortlist candidates which have similar features at these distance, two acceptors and one aromatic. So this is called a pharmacophore based ligand design. So it identifies what's the common features or common pharmacophores present from a list of known inhibitors, and then using this pharmacophore we can go and look into databases to see whether there are molecules with the same pharmacophore, and hopefully if they have this pharmacophore, we believe that it may have the same activity, because ultimately a ligand goes and binds to a protein at the active site. So there is an interaction between the active site amino acids and the structural features of the ligand. So it could be an aromatic type of interaction, or a hydrophobic interaction, or a hydrogen bond donor acceptor complementary interactions. So that is what is called pharmacophore based design. So when we did the pharmacophore elucidation for all these molecules, we find this interesting feature: aromatic to acceptors. So one could go, like I said, go into databases to look for molecule with similar feature. Okay, when you look at the other two proteases, I said, which are involved in the multiplication of the viral RNA, the 3CL protease and PL protease. Again, there are some inhibitors reported for inhibiting this 3CLP, as you can see here. When we do a pharmacophore model for known inhibitors, so we find that all of them have these type of features: two aromatic groups and two acceptor groups at these distances. You can see the distance. Okay, so it's got two aromatic, two acceptors. So like I said, again, if I go to databases and search the databases which have these type of features at these distance, if I find some molecule, I can hypothesize that they also may have inhibiting activity against 3CL. If you look at the inhibitors for PLP, that is the papain-like protease, again there are some reported molecules. Again, if you do a pharmacophore modeling, we find three acceptors and one aromatic. Okay, at these distances. So you can see the PLP is more hydrophilic when compared to the 3CLP, because 3CLP has only two acceptors. That means two nitrogen or oxygen and two aromatic groups, whereas this has got three acceptors. and one aromatic. This is more relatively more hydrophilic when compared to 3-CLP. So we can look at molecules which have overlapping on both these proteases, and if we can find out some new inhibitors which will have pharmacopor overlapping on both of these, we may be able to come up with candidates which may have dual inhibiting activity. In fact, we just our paper got accepted in biomolecular and dynamics of the work which we have done, where we identified candidates which seem to have a dual inhibitory activity binding to both this as well as this protein. So this protein is relatively more hydrophobic when compared to this protein, which has got 3 acceptors, whereas this has got 2 acceptors and 2 aromatics. So we can start with the pharmacopore-based approach if we know inhibitors to identify new ligands, and then we can go into docking-based approach once we have shortlisted possible candidates and go and dock them to the active site. And as I said again, many of the proteins are still not crystallized related to SARS-CoV-2. Only few have been crystallized, and the PDBs are available in the protein data bank. So many of them we may have to take it from SARS-CoV and then do something called a homology modeling. Okay, so there are antiviral drugs which go and inhibit the RNA polymerase, viral RNA polymerase, which is involved in the replication. Remdesivir is one candidate. You must be hearing quite a lot about this in the past couple of months. I think some countries have started using this as a therapy as well. Okay, so different types of known antiviral drugs, okay, they are involved in the inhibitors for RNA polymerase. Okay. Of course, there are other treatments that is not part of the drug discovery, like plasma therapy. Previous speaker must have spoken, interferon beta treatment, all these cell therapy, all these are not drug design approach, but my focus is more on the drug design. So when we talk of drug design, it's more of a small molecule which go and inhibit or stop the action of some proteins or enzymes and so on. So these antiviral drugs have RNA polymerase inhibiting activity, and many of them are known to have that type of activity. Natural products also have been very successful in addressing this SARS-related coronavirus. Many phytochemicals, you can see, imodin, which is found in aloe vera, quercetin, which is found in green tea, licorin, which is found in licorice, licochalcone, again found in licorice, curcumin, found in turmeric, glyverinic acid found in licorice, sucrose. All these have anti-SARS-CoV-1 activity. Okay, when I talk about SARS-CoV, we must assume it's still SARS-CoV-1 activity. New molecules are still not being, as of now, screened for SARS-CoV-2. Okay, so all these molecules, as you can see, have SARS COVID activity. So this imodin seems to be working on this spike protein ACE2 receptor binding activity. Quercetin seems to work towards the proteases enzymes. Then again licorin, again protease enzymes, and some of them it's still not known clearly, and so on actually. So there is there is a lot of interest in natural products based designing inhibitors. So as you can see, phytochemicals, many of the phytochemicals have this type of inhibiting activity against many of the proteins which I showed you actually. So the corresponding plant extracts in which these natural products are found also will be having the antiviral activity. If you have pure compounds, then it is easy for you to understand the mechanism as well, in addition to getting the activity studies. So I mentioned that there is a tool which, if you are interested, can make use of, and it's this is the Webserver. So if you give a inhibitor or a ligand or a molecule, it goes and docks to several proteins found in COVID-19. I have said 49, but as of now, there are almost 77 possible targets. So also it goes and binds to several human targets, and then it ranks them based on the binding energy. So you may find SARS-CoV target in the first rank or second rank. You may find human target, and then it also tells you what is the mechanism. That means it gives you what is the target name. So this is quite an useful server to look at for those who are interested in doing some bioinformatics, because currently it also gives you all the proteins and targets related to COVID-19. It also does a virtual screening. So given a molecule, based on certain features, it tries to predict what could be the targets in the COVID-19. Okay, so I'll show you an example. So this is the web server given here. So what we did was we gave a query molecule, remdesivir. This is currently the antiviral drug being looked at in many countries as a possible treatment strategy. So when we asked it to dock against COVID-19 as well as COVID-19 targets as well as human targets, it arranges them based on the binding energy or binding score. So as you can see, as you can see, it's got a minus 11 binding energy. This is a human target, and then it's got minus 10 binding energy against SARS-CoV target. So this is an RNA-dependent RNA polymerase. Okay, so it seems to go and bind to that. Then it again finds a human target, then several SARS-CoV target and human target. Okay, so what it does is it goes and docks to all the possible which is stored in the database related to human and COVID target. Then it arranges them in the odd ranking based on the binding energy. So it also does, as I said, something called ligand similarity or fingerprint based approach. So given a ligand, it will tell what could be the target, most probable target 1, 2, 3, and so on. Okay, and based on the similarity of the structure with other known inhibitors. So here it has put the RNA-dependent RNA polymerase of COVID as the top because it's got about 60% similarity. Okay, then it tells you the other human targets and so on actually. So as I said, this is a very useful tool, okay, or resource if you are interested in bioinformatics. Of course, there is Swiss target prediction. This also does um, docking based. It will try to predict what could be the possible targets given a ligand. But the current targets it has is only human as well as mouse and rat and so on. So it does not have anything related to the targets of COVID-19. So it's good to use this particular server as of now. As time goes, I'm sure there will be many resources that may be coming in for one to do a lot of bioinformatics. Okay, so the important point you must be hearing nowadays about is the bioavailability and toxicity, because when you design drugs, if it's an oral drug, you need to consider the stability at various pH condition, the stomach pH, which is less than 2 and 4 and 7 and so on. So your drug has to be stable, and then it has to be able to overcome the liver related biodegradation and reach your target site. So many drugs currently that is being repositioned have this problem of bioavailability, and also the toxicity issues are there, because ideally the toxicity should be very far away from your efficacy. So I should be able to give higher concentration without getting any toxicity. But in reality, what happens is as you activity increases, toxicity also increases. So that is why, as of now, many clinical trials that are being done on existing molecules, we find that many of them have this type of behavior. That means the efficacy increases, so the toxicity also increases. So we need to keep that point also in mind. Actually. Okay, so because the coronavirus is a disease which is caused in the upper respiratory tract and pulmonary region as well as in the lungs, why not think about inhalable drug delivery systems? That way, the drug may directly go and reach the virus that's accumulated in the upper respiratory tract and so on. There are not many inhalable drugs as of now available. Of course, for treating asthma and also some congestive obstructive pulmonary disorder, there are some drugs which have been approved by FDA, but there are not many drugs. Even tuberculosis drugs is given as an oral drug. Okay. So the inhaling drugs are given as a one-shot, fixed dosage, and direct drug inhalers. They are given as pressurized metered inhalers. Then they are given as dry powder inhalers and nebulizers. Actually, so the issues of inhalable drug discovery is a little bit serious problem because exposure of high dosage of drug when the drug is given as one shot. So it may lead to local toxicity. For example, if you have very strong drug concentration reaching the pulmonary region, there could be some local inflammatory issues. Higher chances that inhaled drug might be expelled out because of exhalation, cough, or sneeze. Okay. Okay, there could be degradation of these drugs by the enzymes which are present in the lung. So all these issues or points needs to be considered if one is interested in designing inhaled drugs. But the main point is the drug may directly go and reach the required target site, unlike here a oral drug or a drug IV related drug. So you may have to design polymeric systems which may degrade, reach the target site, bind to the target, and then slowly get degraded and allow the drug to get slowly released. So that's what we need to consider if you are designing inhalable drug delivery system. Okay, so to conclude, this is caused more by SARS COVID-2. Okay, although it's got much lower genome similarity, and it has got a higher genome similarity with BAT, it is considered SARS-CoV-1 because the mechanism of action and protein similarities are much closer. Okay. The current antiviral drugs are being repositioned, but one needs to consider the toxicity issues as well. Then I showed you many possible targets: the spike protein, the ACE2 spike ACE2 interacting domains, then DMPRSS2, then
इस
glycine.
बार आप कल बोल रही थी ना कि इस बार शायद चाहिए
Then we have the natural products, extracts, and
अलग
pure phytochemicals I showed you. Okay, currently only few targets are available in PDB because only few have been crystallized. Okay, so as time goes, many PDBs will get
मुझे लगता है इसका
crystallized.
तो
So
ज्यादा
enable
है
can have
क्योंकि
better
प्रोजेक्ट का तो एक स्टूडेंट चाहिए
efficacy. Okay, so with that, let me conclude my talk. Okay, let me thank my research scholar who helped me to do some of the ligand-based drug design. Let me thank the organizers once again for giving me an opportunity, Professor Venkateshulu and Professor Kripanadi sir, for giving me an opportunity. Thank you very much for your time. Thank you.